The new golden age of immunology. The golden age of immunology was from 1870 to 1910. This is when the basic elements of immunology was discovered, as well as important vaccines. The new golden age, we have seen 75 new vaccines being developed, some for AIDS, malaria, and cancer. We've seen improvements in old vaccines using adjuvants. Adjuvants are chemicals that may increase the effectiveness of response to an antigen in a vaccine. So it improves the effectiveness of some antigens. We have seen improvements in delivery, so a pressure gun, in food, in patches, etc. And of course, we've seen safer vaccines. Diagnostic immunology. The first diagnostic immunology test was the test for tuberculosis, and Robert Koch did this. Diagnostic techniques help us to determine the etiology of disease. Examples of diagnostic techniques: microscopy, culture by testing the biochemical properties of the microbe, molecular, using a PCR to amplify a gene associated with the disease and identifying the gene on a gel. Immunological, how big is an antibody? Can we see it under a microscope? Diagnostic immunology involves using the principles of the immune system or antibody-antigen reaction to diagnose diseases or detect antigens in bodily fluids. There are 5 important diagnostic tests: direct agglutination, indirect agglutination, hemagglutination, both regular and viral, direct ELISA, and indirect ELISA. The principles of diagnostic immunology. The physician collects a sample. There are 2 types of samples. The first one is an antigen sample. The antigen sample is a bodily fluid that contains the infected microbe or the microbe's toxins. So it may be present in urine, feces, blood, skin, pus, throat swab, mucus, etc. Blood antiserum sample contains the antibodies that the patient made against an infection. If the patient is infected with the suspected pathogen, then his or her serum has those antibodies in it. If the sample is antigen, then the physician exposes it to pre-made antibodies for the suspected pathogen. The antibodies are produced by a rabbit that was infected with that organism. They are collected in sterile vials and sold by pharmaceutical companies. The blood antiserum sample, then, physician exposes it to an antigen from the suspected pathogen. The antigen from the microorganism is prepared by the pharmaceutical company. It could be a toxin, an inactivated whole agent, or any subunit from the suspected pathogen. The antibody titer is the concentration of antibodies against a particular antigen. So it's the amount of antibody in our antiserum. It can be used to determine how far a disease has progressed. So if you have a lot of antibodies, and one of the things that we learned back in chapter 10 was that, okay, there's a delay in the number of antibodies that's being produced. So we know that antibody levels usually don't peak till when? If you said 2 weeks, you're right, approximately 2 weeks. So if you have very low antibodies, that means that you just started this infection. If you have high numbers, then you know you've had it for quite some time. Direct agglutination tests can be used to detect the antibody titer of a person. So this is just an example of showing the number of antibodies. And of course, we did something like this in the lab with the staphylococcus test, where we saw a speckled appearance, and this is just a regular drop of blood liquid. Okay, that means that it's non-agglutinated versus agglutinated. When a person goes from no antibodies to elevated antibodies, then this is called seroconversion. Agglutination occurs when the antibody binds to more than one antigen at a time. Direct agglutination tests are used to detect antibodies in antiserum that are specific for large cellular antigens. So it's used mostly to determine titer and to determine the stage of the disease. Indirect agglutination is for soluble antigens. It's used to detect antigens in patient sample or antibodies in the patient antiserum. You have soluble antigens or antibodies that are absorbed onto latex beads. So this is also known as a latex agglutination test. This type of test is very fast, it's rapid, and we've seen this with a 10-minute strep A throat test. You go into the doctor and you're like, I have a sore throat, and the doctor's like, okay, I think you may have strep. And he tells you to open up wide, and he takes a cotton swab and he swabs the back of your throat. That throat swab sample is the antigen, okay? And it's the antigen is going to be Streptococcus A if you have strep, okay? And he's going to expose that throat swab sample to the antibody in the kit, in the test. And if you do have strep, then it's going to agglutinate with each other and provide a positive result. If you don't have strep, then remember, antibodies are specific. They're not going to bind to anything else, so your test will come out negative. Okay, so that's how the strep test works. So here's a picture, and this here that you're looking at here represents the strep test here. So you see the latex beads with the antibodies, and these are your throat swab samples. Hemagglutination involves the agglutination of red blood cells. Blood is the antigen in the agglutination test. This helps us to determine blood type. If the person has type A antigens, then it will agglutinate in the presence of type A antibodies. So blood cells are agglutinated due to antigens that are present in the blood. So viral hemagglutination. In viral hemagglutination, some viruses agglutinate red blood cells in vitro. So mumps, measles, and influenza will do this. So when we say in vitro, inside of the body. So we have something called the viral hemagglutination inhibition test. The antibodies bind to the virus first, inhibiting the clumping of red blood cells when blood is added. So we're going to take the serum sample from the patient and mix it with the suspected viral pathogen. We're going to add the blood. So here, in a normal circumstance, measles, mumps, or rubella, they'll take your red blood cells and they will hemagglutinate them. They will agglutinate them. However, if we take your red blood cells and then we add the antiviral antibodies, remember one of the functions of antibodies is neutralization, right? And we said neutralization is what? Right. Neutralization means that it's going to inhibit it from being able to bind to our host cells. So in this situation, the virus will not be able to bind to the red blood cell. And we have neutralized hemagglutination. Okay, so the antibodies have prevented hemagglutination. ELISA stands for enzyme-linked immunosorbent assay. The enzyme reacts with a substrate to produce a colored product. In the direct ELISA, the enzyme is linked to an antibody, of course that's made by the pharmaceutical company. It detects the antigen from the patient sample. So a good example of this is the home pregnancy test that's used to test for the HCG hormone. HCG hormone is a protein present in the urine when a woman is pregnant. So this serves as an antigen. And of course, in that test is the antibodies, and if they hook up, you're pregnant. Indirect ELISA is enzyme is linked to an anti-HISG, which is an artificial antibody that reacts with any human antibody that is already bound to an antigen. So its job is to detect antibodies in a patient's serum. And this is exactly how the home HIV tests work. And you'll see here that this is a website that you can go to that talks about the quick oral HIV test at home, and you could go there and you can get information about how this test actually works. So here is a picture of the direct ELISA, and this was a picture showing you the pregnancy test. right? So there's already in that test an antibody. So the woman pees on the stick, and if she has the hormone present, then it would bind with that antibody, and then you will have a color change. And that color change will tell you that you're pregnant. And then this is an example of the indirect ELISA. And inside of this test is the antigen, HIV antigen. And then basically you would put your oral swab into the kit. And if you do have antibodies specific for that HIV, then it will give you a result. Here ends Chapter 11.